Welcome to BioVivacious. BioVivacious, as you know, is a YouTube channel dedicated to clear fundamental concepts of biosciences. Today we are going to discuss about polysaccharides. Polysaccharides, as you know, when more than 10 monomeric units of carbohydrates, when they join together, it becomes a polysaccharide. Now, which means when a polysaccharide is hydrolyzed, it will give its monomeric units. If a polysaccharide is made up of glucose, if the monosaccharide units that you will get after hydrolysis will be glucose. If it is made up of fructose, if the monomeric unit you will get it after hydrolysis will be fructose units. This is a polysaccharide. One of the interesting aspects of a polysaccharide is polysaccharides can be branched, which is very unusual of a biomolecule. You look at other biomolecules like proteins or nucleic acids, they cannot be branched, whereas carbohydrates can be branched polysaccharide. So if a polysaccharide is made up of only of glucose or only of fructose, we say that it is a homopolysaccharide. So what does that mean? It means you will find only glucose units inside, or you will find only fructose unit inside, which is called a homopolysaccharide. If it is made up of more than one monomeric unit, it is called a heteropolysaccharide. Okay, so homopolysaccharides examples are starch is an example of a homopolysaccharide, glycogen is an example of a homopolysaccharide. Example of a heteropolysaccharide, I'm sure you heard about mucopolysaccharides. That is an example of a heteropolysaccharide. Now, if it is only made up of glucose, we call it a glucan. If it is made only of fructose, we call it a fructan. So therefore, you will find such varieties of polysaccharides present in nature, doing varieties of functions. Some are involved in the structural integrity of a cell, some will act as a storage molecule, and some polysaccharides will also act as a signaling molecule or a recognition molecule or as a receptor. So as part of a receptor. So these are the varieties of functions polysaccharide performs. So let us look at some of these polysaccharides elaborately. We will begin by looking at storage polysaccharides. In the first storage polysaccharide we are going to look at is starch. So starch is a homoglycan. Starch is a homoglycan made up of exclusively glucose units. So it has only glucose present. So when you look at the starch, so a starch is made up of two kinds of polysaccharide. Which are the two kinds of polysaccharide? You will find something called amylose. It is one of the polysaccharide which is present in starch. Another kind of polysaccharide is amylopectin. So these are the two kinds of polysaccharides found in starch. There is something very interesting about amylose. See, we have seen that it is made up of exclusively of glucose units. Now, how these glucose units are linked together? They are linked together by alpha-1,4 glycosidic linkage. Let us simply draw. So this is CH2, one glucose unit. So this is the alpha D glucose. Another alpha D glucose is drawn here. So therefore, if the linkage is between the first and the fourth carbon atom, this is 1, 2, 3, 4, 5 and 6. 1, 2, 3, 4, 5 and 6. So the linkage is between this first carbon atom, this first hydroxyl group with the fourth one, with the release of a molecule of water. So a molecule of water is removed, so therefore it becomes a residue. So this is the basic structure of starch. So this can repeat n number of times, this basic unit. So joining one after the other through alpha 1 4 linkage. So this polysaccharide is what we call amylose. This is the structure of amylose. So you will find many thousands of units of glucose joined together through alpha 1 4 linkage. So therefore the molecular weight of amylose can be about 500,000. That can be the molecular weight of amylose. Remember, amylose is a relatively soluble portion of a starch in comparison to amylopectin. So when you put starch in water, this is a portion that will absorb water and it will start dissolving. You see that the color changes. Especially when you cook rice, you will see the color of water changes. Okay, that is because starch comes or getting solubilized in the hot water. Now this is amylose. What is the structure of amylopectin? Amylopectin has two kinds of structures. One is alpha 1 4, similar to this, and the other one is alpha 1 6. So therefore, you know that this is the sixth position. Alpha 1 6 means it will create a branching. So therefore, if you keep on assembling, these are the amylose unit. How to make amylopectin? Draw an alpha 1 6 linkage. So this becomes amylopectin. What is interesting is how often these branches occurs. In the case of starch, these branching occurs once in maybe about 15 to 25 residues. After about 15 to 25 residues, you will find the branching. So therefore, amylopectin is when we talk about starch, we are saying that it is a highly branched structure. See, this complex, this polysaccharide also gives a very good advantage. Remember where is starch synthesized? It is synthesized in leaves. Okay, and it is synthesized in leaves, and it will be in the chloroplast. Okay, so and it is stored there, and from there, after it is synthesized, it will be transported to various places. To maybe it will be stored as a tuber, it will be stored in the grains. Okay, so in varieties of places, starch will be stored. Now this polymeric form also has one big advantage. The biggest advantage is, if you look at this structure, you will see that which is the reducing end. This is the reducing end. This is the non-reducing end. Non-reducing end of a starch. So therefore, a starch molecule will have plenty of thousands of non-reducing end. That means that if the starch cannot be easily degraded, it will not undergo unwanted reactions because only the non-reducing end is available. So therefore, this can prevent unwanted reaction. Not only that, there is another big advantage of storing starch as a polymer. Starch can minimize the osmotic pressure. You know that osmotic pressure depends on the number of molecules that are present. So compare a starch made up of 1000 glucose molecules with 1000 individual glucose units. So therefore, a starch of 1000 glucose units will insert or 1 by 1000th osmotic pressure. That is another big advantage of storing biomolecules as a polymer. So let us look at some of the properties. So starch, as you know, it's a white powder. Now this white powder, we have spoken already about the solubility of starch. Now if you look at the amylose structure, see the amylose structure, it has the tendency to form micelle structure. both amylose as well as amylopectin can form missile structure. So amylose forms a missile structure and about 6 residues form a helical structure. 6 residues form a helical structure, and one of the very important tests for identifying starch is to add iodine. Normally iodine gets embedded here and that will give a blue color to amylose. Whereas in the case of amylopectin, the inner core is normally not digestible. So it can take up water, it can swell. So, and this amylopectin has the tendency, it will react with the iodine and it gives a slightly purplish color. You will be experimenting in the laboratory how these colors are formed. You will be able to see that. Let us look at how exactly starch can be degraded. Starch is degraded with the help of 2 enzymes. In the 2 enzymes are, one is alpha amylase and other one is beta amylase. So these are the 2 enzymes that are required for degrading starch. If you talk about alpha amylase, which is present in our saliva, which is also present in all animal systems. So these 2 units join together and we have structure of starch. In the salivary alpha amylase, it can degrade starch. Remember, this is an endoamylase. What is the meaning of endoamylase? It will act inside the structure of starch. It will act inside the structure of starch. So this endoamylase can cut the starch into dextrin or it can cut into maltose, etc. So therefore it is an endoamylase. And it will cleave specifically alpha 1,4 linkages. What about beta amylase? Beta amylase is an exoamylase. Exoamylase means it will act at the non-reducing end of starch. So what will happen if it is acting on the non-reducing end of starch? It will release especially maltose or glucose units. So that is how these 2 enzymes act on starch. What will happen when there is an alpha 1,6 linkage? So amylase cannot act on it. So therefore we need a 3rd enzyme, that is alpha 1,6 glucosidase. So alpha 1,6 glucosidase can cut in the alpha 1,6 linkage and remove the branch of starch and make it glucose units or make it a linear molecule. So then again amylase will act on it and it will cut, remove maltose or remove glucose unit. That is how starch is degraded in every living system. So we have seen a very brief outline about the structure, where it is stored, and some of the properties, especially with enzymes.